Hello, good evening. Let's wait for 2 minutes more, then we'll initiate. Okay. Okay, so let's talk about today's session. Okay, so today we'll be studying about the immunotherapy. So here we'll be talking about the cancer cells and how immunotherapy is being used to treat those cancer cells. Okay, basically we'll be studying about the CAR T cells. Okay, so CAR T cells are a modified or a genetically modified T cells. Okay, so we know that our body has these T cells, B cells, which are the lymphocytes, okay, that fight and give immunity to our body. Okay, it fights the infection. So similarly, there are T cells in our body that fight different kind of infection. Okay, that also includes the cancer. Okay, so today we'll discuss the genetically modified T cells, which are referred as CAR T cells. Okay, so this CAR is chimeric antigen receptor T cells. Okay, so first, to know what are the CAR T cells, we have to know what are the T cells and how does they function. What is their function? Okay, so T cells are the immune cells of our body. Just a minute. Just give me a minute, please. So, a T cell is a type of immune cell that is having receptors on its surface Okay, and this receptor can identify the antigens. Okay, so suppose this is a infectious cell that has some antigen or that expresses some antigen on its surface. Okay, and these T cells will be having receptor for that particular antigen. Okay, so when there is a binding of the surface antigen with the T cell receptors. Okay, okay. So then there are signaling processes that activates the T cells. So T cells get activated. Okay, then they will divide, multiply, divide, and release the chemicals referred as cytokines. Excuse me for a minute, please. T cells, which is having receptor on their surface. Okay, and these receptors identify the antigen which is presented by the this cancerous cell. Okay, and this recognition or binding of the antigen and receptor uh, activates the T cells. Okay, gives the T cell signal for activation, and after the activation this T cell gets divided and then release the cytokines, the chemicals. Okay, which will kill these infectious cells. Okay, and then yeah, and which will kill these infection cells, and also it becomes it remains in our circulation as a memory cells. Okay, so that now when the further infection happens, then also these cells will behave as a memory cells and fight against those viruses or infectious cells. Okay, so these are the T cells, normal T cell function. Okay, so now scientists wanted to utilize these property of the T cells, okay, for the specific recognition of the antigen molecules, and then killing them, and also make a memory for them in order to prevent the further infection. Okay, so there they modified these T cells. So generally what happens, okay, so a normal T cell has a receptor like this, a receptor like this. Okay, so this has a binding domain. Okay, a transmembrane domain, transmembrane domain, and this you can say a signaling domain. Okay, so uh, these binding domain identify or binds to the antigen, but the limitation here is these T cells or the receptors, T cell receptors, which I'll refer as TCRs now. So these T cell receptor bind to an antigen only if that antigen is being presented by a other cell by a MHC molecule. Okay, this is a major histocompatibility molecules. Okay, so Suppose if some other cell, okay, that is having this MHC molecule, it is presenting this antigen with the MHC molecule, then only the T cell can identify this antigen and bind to it. Okay, so that is one limitation here. And once the binding takes place, then here the signaling molecule gets activated. So this is the first signal. Okay, so this is the first signal which gets activated. Okay, and then furthermore, there is one more binding happens. Okay, which is a which is a, you can say, a co-stimulatory signal. Okay, it is a co-stimulatory signal. So this is signal number 1 and this is signal number 2. So after these two signals and these two bindings, only the T cell activation takes place. Okay, so here in the modified T cell or the CAR T cells, what they are having here is, I'm making it here. So scientists genetically engineered such a T cells, okay, which is first the limitation was this, they will only bind to those antigen which is presented on the MHC molecule or via the MHC molecule. Okay, so to limit that, what they wanted to make is they can identify any antigen. Okay, so the receptors should bind to any of the antigen, whether it is presented by the MHC molecule or not, to avoid the limitation of any extra cell being involved in this binding or recognition process. Okay, so what they have done, such a portion or such a biomolecule that can bind directly to the antigen is an antibody. So uh, the the antibodies, okay, so they have taken half of the portion of the antibody, okay, particular antibody, so that the recognition process will be easier. It will directly bind to the antigen. Okay, and then to combine the first signal and the second signal, they have fused this signaling molecule and this signaling molecule. So this is CD3 zeta and this one is CD28. Okay, so they have fused these two and made a single, or infused in a single receptor, transmembrane receptor. Okay, zeta and CD28. Okay, so these two are been combined, and to join it with the antibody portion, they have made a transmembrane portion also, which has taken from some other molecule. Okay, so this is now a CAR T cells has been prepared, which is chimeric antigen receptor T cells. Okay, these are CAR T cells. So they kind of made a chimera, fused two or three proteins all together to make a single transmembrane receptor on the T cells, okay, or that expresses on the surface of T cells. So here, this particular portion which is derived from an antibody, then a transmembrane portion, okay, and here the uh, signals, the first signal and second signal, those particular molecules combine. Okay, so these are the signal that are uh, that recognizes the specificity or the type of antigen and accordingly activates the T cell to release certain kind of cytokines that will kill those infected cells. Okay, so for the genetic modification, so the genes encoding each of these portions have been fused together, in a DNA, and DNA that DNA is in uh, delivered via a viral dead virus into the T cells, and there it got expressed and expresses on the surface of the T cells and becomes a modified CAR T cell. Okay. So here you can see the CAR transgene. Okay. It will be referred as transgene because it is a fusion of three different portions. So CAR T cells are typically produced by transducing T lymphocyte with the transgene encoding a synthetic antigen receptor, which is a chimeric antigen. Okay. So this transgene is integrated into the T cell genome, transcribed and translated into a CAR protein. So the binding and signaling domains separated into extracellular and intracellular compartments. So this is the binding domain, the extracellular compartment, and this is the signaling domain and the intracellular compartment. So the extracellular domain is derived from the variable region of an antibody that recognizes specific tumor antigens, together with a spacer that provides flexibility to this particular domain. So this is the CAR T cell receptor structure. Okay. So yeah, so there are several kind of, so this co-stimulatory molecules are the different molecules that have been used according to the different response or different type of cancer cells. Okay, so there are different generation of these CAR T cells. So the first generation of CAR T cells contain only CD3 zeta as a well-documented intracellular signal domain. So in the first generation of the CAR T cells, only CD3 zeta were there. No second signal was there. That is why, to make it more efficient, both the signaling proteins are being fused. CD3 zeta and also CD28 or 41BB depends on the type of cancer cells that has been targeted towards it. Okay, so second and third generation of CARs involve one or two co-stimulatory molecule. Here you can see in the second generation one co-stimulatory molecule, in the second generation two co-stimulatory molecule. Okay. As well, the fourth generation of CAR T cells strongly motivates the downstream transcription factor to promote cytokine generations. Okay. So here in the fourth generation, along with all these modification, there are certain modification that allow the overexpression of cytokines so that the cytokine release can kill the cancerous cell in a faster manner. Okay. Okay. So prominently, the genome addition technologies such as CRISPR-Cas9 have been widely used to construct the um, track TCR deficient and CAR T cells, establishing a fifth generation of CAR T cells. Okay, so in the fifth generation, what is the concept here is this particular T cell also have a these MHC molecules, which are TCR alpha beta transmembrane proteins. If they are produced or expressed on the these modified CAR T cells, they will cause the alloreactivity. So to avoid that alloreactivity or the reactions, these genes or these are the tracked genes that have been knocked out from the modified CAR T cells, and that is why they are not able to express on the surface, and that is why it becomes a more efficient CAR T cell. Okay. So these are the most commonly used target for CAR T cell therapy are surface antigen that has been used in clinical studies. Okay. So these are the surface antigens that has been used, and these are the type of cancer. So this is the list you can remember. So carcinoembryonic antigen as, is used as a surface antigen for the colorectal adenocarcinoma. Okay. Similarly, IL-13 receptor alpha used in a glioma type of cancer cells. So this is a table that you can remember for the further questions answer. Okay. So there are also some challenges which needs to be overcome. Okay. So as I have mentioned earlier, that these kind of receptor on the T cells that causes the uh, autoreactivity or alloreactivity. So before talking about that, certain limitations. So how this CAR T cell therapy works? So if a patient is being diagnosed with the cancer, to treat him with the CAR T cell therapy, what are the procedures? Is the first step will be to um, take a T lymphocytes or the lymphocyte filtrate from the patient. Okay, so first, when the patient gets detected with the disease, to treat it with the CAR T cell therapy, you need the lymphocyte so that you can culture it and make it genetically modified. Okay, so the T cell lymphocytes from the so the blood is collected from the patient, and from there the T lymphocytes are only sorted and filtrated. Okay, so after they are filtrated, the second will be culturing them. Okay, culturing, and then third step will be uh, introducing a foreign DNA or a transgene. Okay, transgene, introduction of transgene. Okay, and their expression on those modified T cells, and then and make them divide, increase their population, select those particular kind of CAR T cells or transgene expressing modified cells, and then those cells are being put back into the patient's body so that they can detect the specific antigen against a particular type of cancer and kill those particular cancer type cells for the cure. Okay, so uh, back to patient. So the fourth will be growing the CAR T cells. So this entire duration takes approx 1 to 2 months. This entire work takes approx 1 to 2 months. So, and this is only for specific for one particular person. So to simplify this particular technology, what scientists are aiming to do is they aim to make a universal CAR T cells. Okay, so that uh, when a patient comes for the CAR T cell therapy, there is no need to take out his own lymphocyte and modify them and put it back into them. There should be a stock ready of the CAR T cells that can be directly uh, infused in the patient's blood so that the patient can be treated more quickly. Okay, so those are the limitations. So to overcome those limitations, uh, these universal CAR T cells are being designed. Okay, so these universal CAR T cells, if like some one patient's CAR T cells or T cells is being given to another person's body, then there are chances of the graft rejection. Okay, okay, post graft rejection. Okay, because of the presence of certain antigens that that are being recognized in somebody else's body as a antigen. So what is being done here is the I explained you, these are the surface markers that work as a foreign antigen or the allo antigen. So these particular gene that expresses those particular surface antigens are being removed by using the CRISPR-Cas9 technologies, so that the particular CAR T cells expresses only one kind of T cell receptors, which is chimeric antigen receptors, and only being used to cure the disease. Okay, so yeah, so that is what call. So to avoid the autologous CAR T cells, the off-shelf allogenic CAR T cells, these have the potential advantage, including immediate availability. As I mentioned, to avoid the delay in the cure, the immediate availability of the cryopreserved CAR T cells for the patient in the urgent treat, and enough quantity for the first infusion or redosing, and possible standardization of CAR T cells producing. Whereas considering the presence of endogenous HLA or T cell receptor on donors T lymphocyte, the biggest challenge of universal product is the potential risk of alloreactivity. Okay, host versus graft response and graft versus host disease. Okay, so genome editing has been widely used to generate the off-the-shelf allogenic CAR T cells that remove the alloreactivity and address the other limitations associated with the immunotherapy strategies like TCR T cell therapy. Okay, so off-the-shelf T cell product, it increases the histocompatibility between the donor and the recipient cells and prevent the rejection of T cell by the host, and inhibit the alloreactivity of the product so that the adoptive transferred T cell do not exert a cytotoxicity against the recipient cells. Okay. So anti-tumor alloreactive receptor expressed on the chimeric antigen receptor and T cell receptor engineered T cells. So three possible TCR complexes on the TCR engineered T cells. Okay. So here the first one is the endogenous TCR may exert a alloreactive function. So as I have mentioned, the endogenous this T cell receptors that expresses on the surface of the T cells is they will cause a alloreactive function. Okay. So here this is the TCR alpha and beta subunit, CD3 zeta, CD3 delta, CD3 zeta, and all these different kind of subunits. These are all are potential. They will all cause the potential alloreactivity. Okay, and the second one is the transgenic or TG T cell receptor containing transgenic alpha and beta TCR subunit is responsible for the anti-tumor activity. This particular one, it is a mixed TCR which work as a universal kind of T cell receptor where the transgenic alpha and beta chains are made here. And the third one, it is also possible that a combination of TG and an endogenous subunit forms a mixed TCR containing that result in potentially undesired function. So this is also can be made, which causes an anti-tumor activity. Yeah, so this is the second one, sorry, anti-tumor activity here. And this is a potential unwanted activity, which is a mixed TCR. Okay. So these are the modification that can be done. So yeah, so these are the cells or the genes I talked about. So genome editing technology such as zinc finger nuclease, TALEN, and CRISPR-Cas9, all that can be applied to knock out the track, which are T cell receptor alpha constant and T cell receptor beta constant loci to prevent the TCR expression and alloreactivity. So as this one, which causes the alloreactivity, so the TCR alpha and TCR beta genes needs to be knocked out from the CAR T cells so that it does not express and causes an alloreactivity. So this is the T cell, and here the CRISPR machinery, zinc finger nuclease, TALEN, or CRISPR machinery, anything can be utilized. So in the zinc finger nuclease, the zinc finger domains along with the FOP1 enzyme makes a cut and disrupt the locus via NHEJ or the HDR mediated method. Similarly, the TALEN is also being used in the similar manner with the FOP1 endonuclease, or to be a more specific and more convenient manner will be the guide RNA guided a Cas9 endonuclease, which makes a cut in these particular genes, which are alpha and beta subunits coding and other cytokines producing or MHC1 or 2 producing transmembrane proteins on the surface of the T cell receptor, so that they will create a break, disrupt the locus, and similarly here you can see knockout of the genes such as B2M, TAP1 transporter associated with antigen processing, transporter associated with antigen processing, and TAPSIM involve MHC1 and 2 with the universal histocompatibility. So all these modifications can be done if the CAR T cells are fused or are also utilized with the CRISPR-Cas9 or the TALEN machinery. Okay. It's also the similar, the CRISPR knockout or knockdown, and also there will be one more modification can be done, that um, to enhance the cytokine production. Okay. For cytokine production enhancement also, there are certain modification can be done using the this particular CRISPR-Cas9 machinery. So here you can see that this particular uh, CAR, uh, CAR antigen, which is produced here on the membrane of the T cell, which includes a CD3 zeta, which is for signal 1, the co-stimulatory factor, signal number 2, fused with a transmembrane and joined with a binding domain, which is the uh, produced of the antibody that specify or that recognizes specific antigen, which is expressed on the tumor cells. Okay, so this is a tumor cell and it is expressing one particular tumor antigen, which there is no need of presented by the MHC1 by some other cell. It can directly recognize the tumor antigen of the tumor cell. Okay, and this is the intracellular TGF beta, the TCR receptors, which can be knocked out using the CRISPR technology, and also these particular receptors which shows an inhibitory effect, or these tumor cells have these particular um, uh, proteins that interacts with these receptors, for example PD1 and other two, to suppress the action of the T cells. So these PD1, CTLA4, TIM3 all can be knocked out so that there will not be a off switch for the activation, T cell activation. Similarly, to enhance the cytokine modulation also, CRISPR-Cas9 based gene editing combined with viral or non-viral DNA delivery permits simultaneous biallelic or sequential gene targeting to engineer T cell with expression cassette in site-specific manner. Okay, cytokine encoding DNA cassette can be knocked in, okay, to increase the cytokine production. Into targeted genomic locus, placing these genes under the control of specific promoters for temporal control of expression. For example, IL-15 has been knocked out into the IL-13 locus. Okay, so one more advantage of using the CRISPR-Cas9 machinery is, so yeah. Yeah, so one more reason to use CRISPR-Cas9 is, as I mentioned in the beginning, that the T cells are modified by a lentiviral DNA that is transfected to the T cells, and these lentiviral, so here suppose you have provided a viral, your transgene with a viral DNA, okay, it will go into the nucleus of the T cell and get integrated, okay, get integrated with the T cell genome. Okay, so there this transgene is present, and this transgene get expressed. This transgene can expressed and expressed on the surface of the T cell, become a CAR T cells. Okay, but there are non-specific integration also. So if the viral DNA is being used, the integration of the viral DNA to the genome of the T cells is not regulated. It can integrate at any place. So if the integration of the transgenes happen at the under the promoter that is a very strong promoter, so this particular very strong promoter increases the production of these T cells, or the transmembrane receptors, okay, the modified transmembrane receptors. So these T cells also activates the other immune cells, okay, and that immune cells also release cytokines. So there will be a lot of cytokine release due to the presence of these kind of highly expressed CAR T cells. So, and that increased production of the cytokines can also, like, it will give a mild symptom for a mild fever to a organ failure as well. So in order to regulate that, uh, there will be the CRISPR-Cas9 based method or gene editing can be used to put this particular transgene under a promoter that is having a modular expression levels, okay. And yeah, so here also you can see, like, as I mentioned, the cytokine encoding DNA cassette can be knocked in and integrated into the genomic loci. So for example, so this IL-15 has been knocked into the IL-13 gene locus, displacing IL-15 expression under control of the endogenous IL-13 promoter, as IL-13 is highly active upon T cell activation. So in order to avoid the high activity of T cell activation, these particular genes are placed under a certain promoter, so modulate their expression upon D cell activation. So this creates an inducible T cell specific IL-15 activation switch. Okay, so IL-15 acts as an immunomodulatory in cancer treatment by inducing the differentiation and proliferation of T, B, and natural killer cells. So so yeah. So the further technologies also involve the identification of TAA or tumor associated antigens, or called neoantigens. Okay, tumor associated antigens, or the or new antigens. Okay, so new antigens are those antigens when a particular cells transforms into a cancerous cell. If you do a whole proteome analysis of the cells, there are certain antigens that are being mutated in those particular cells, and due to that it becomes a cancerous cell. So those particular antigens are referred as new antigens or tumor associated antigens. Okay, so how to identify these particular antigens? So if you can identify these tumor associated antigens or neoantigens, you can modify your CAR T cell receptors according to that antigen to make a more specific response. Okay, so both tumor cell and tumor infiltrating lymphocyte are isolated from the patient. Okay, tumor cells and also the T cells, both are isolated from the patients. Okay, so the isolated tumor cells are subsequently subjected to the gene sequencing. Okay, so these tumor cells are subjected to the whole genome sequencing. Okay, and also the mass spectrometry analysis, and via the further bioinformatic analysis method, uh, and biometric analysis promoting the identification of tumor specific uh, tumor associated antigen or new antigens. Okay, so after the identification of those tumor associated antigen or new antigens, these particular clonal neo antigens are uh, uh, grown together with the T cells. Okay, so to validate the immunogenicity of the identified neoantigens, antigen-presenting cells expressing the identified neoantigens are co-cultured with the lymphocytes isolated from this patient. Okay, so this will also validate, like, you are finding from the bioinformatic analysis that these particular genes are being mutated, and you can categorize them as a new antigen, and also those particular antigen presenting cells, you can co-culture with the T cells that is derived from the patient, and that response can also tell or validate your immunogenicity against that particular new antigen. So the specific population of the lymphocyte bearing the and these particular neoantigen specific receptors which exhibit cell proliferation or cytokine secretion in response to the stimulation of the APC expressing tumor specific neoantigen can be isolated. Okay. So after the co-culturing, you can just select those particular T cells that is having receptor against those particular neoantigens. Okay. So this is one receptor, another receptor T cell. Suppose this is another T cells. Okay. So a specific population of TILs, these lymphocytes, bearing a TAA neoantigen. So suppose um yeah. So suppose this one is your neoantigen out of certain proteins. This particular one is your neoantigen. So the specific population of TAA or neoantigen specific TCR. So out of these three, this one is the specific TCR for this particular neoantigen, which also exhibit the cell proliferation or the cytokine secretion in response to the stimulation of this antigen presenting cell expressing the tumor specific neoantigen. So if there is a cell that is this neoantigen is being expressed on that particular surface, and this out of these three cells, this particular T cell which can identify that or which is having a receptor against those neoantigen can be isolated, and this neoantigen specific TCRs can then be cloned further. Okay, so subsequently the cloned TAA neoantigen specific TCRs are transduced into a patient derived T cells, generating a genetically modified neoantigen specific T cells via ex vivo activation and expansion. Okay. So the modified T cells bearing the neoantigen specific TCRs can be adaptively transferred to the patient and target tumor cell bearing the tumor specific neoantigen with high specificity for elimination. Okay. So this is the, so let's finish our question answer. So which of the following is not a correct match for the surface antigen targets for the CAR T cell therapy? This particular question you can do from the table that I have shown in the beginning. The first one, like which particular antigen against which particular kind of cancer. So antigen, and this is the cancer. So the table I have given, so there the carcinoembryonic antigen. This particular antigen was for colorectal adenocarcinoma. This one was the correct option. You can just check the list here. Okay. From the list, you can solve those particular question. I think you can solve. I think the correct answer will be this one, but you can just check. Okay, so what is cytokine release syndrome? So as I have mentioned, those CAR T cells are the highly activated T cells, okay, which can also activate the other immune cell to release cytokine. So a lot of cytokine release can damage your body's own cell. So that is called, that situation is called a cytokine release syndrome in presence of the CAR T cells. So this cytokine release syndrome is the associated with the supraphysiological cytokine production, okay, and a massive in vivo T cell expansion. So the more cytokine production followed by the T cell expansion, that is the cytokine release syndrome, which can damage your own healthy cells as well. So this is the correct option. And this one, elevated cerebrospinal fluid cytokine levels and blood-brain barrier disruption. This is another side effect of the this CAR T cell therapy, where the extra production of the cytokines can also damage the blood-brain barrier and also damage the certain parts of the brain. Okay. Which TCR complex on the TCR engineered T cell is responsible for the anti-tumor activity? Okay. TCR engineered T cell responsible for the anti-tumor activity. So the endogenous TCR is not. So the transgenic TCR containing transgenic alpha and beta TCR subunits. This is this particular one. Okay. The combination of TG and the endogenous subunits will produce a mixed kind of response or unidentified response. And so correct will be this one. Transgenic TCR containing the transgenic alpha and beta TCR subunits, which we have discussed here. This one, anti-tumor activity with the TGTCR. This one. Transgenic TGTCR containing transgenic alpha and beta TCR subunits responsible for the anti-tumor activity. So which of the four was often called the father of cancer immunotherapy? It is Dr. William Coley, which first used the immune cells to treat the cancer. So there the immunotherapy is popular from, where he used a certain kind of bacteria, which is they call Colin's toxin, and that he has used to treat several kind of cancers. So which of the following is not a feature required for a candidate target antigen that are used for TCR engineered T cell treatment? Which are not a feature. They must be selectively expressed in tumors and not in normal cells. They are related to oncogenesis. They are able to evoke T cell response. They must be selectively expressed in both in tumor specificity. Yeah, I think all are required. They must be selectively expressed in tumors and not in normal tissue. Yes, this is correct. They are related. They are able to evoke T cell response. This is also correct. They must be selectively expressed in both tumor and not normal tissue. In both tumor and normal tissue. No, this is also correct. No, but this one cannot be correct, because if you select a particular target antigen which also express on a normal tissue, then it will cause non-specificity, and that is why it cannot be used as a target antigen for the TCR engineered T cell treatment. So which of the following correctly defines TAA? Okay. So this is tumor associated antigens are the peptide that originates from the endogenous wild type proteins. Yeah, they originate, but they are mutated. They originate from the endogenous wild type protein only, but they are the mutated proteins. So they are mutated, the proteins are associated with the tumor. So those are the tumor associated antigens. So their expression is elevated in tumors, that is correct. So limited in magnitude or in spatial expression in healthy tissue. Yes. So in the healthy tissue, they are having limited in magnitude and spatial expression. So their expression is controlled in the healthy tissue. However, their expression is elevated in the tumor tissue growths. And they are endogenous wild type proteins, but they are mutated. And then it is called either new antigen or the tumor associated antigens. The correct option will be all of the above. So which of the following is one of the major methods for adoptive cell therapy? Yeah, use of this T cell receptor modified T cells. The adoptive cell therapy will be the T cell receptor modified T cells. As I have mentioned, there the T cell receptors have been modified, and those are called as the adoptive cell therapy. So genome editing technologies such as zinc finger nuclease, TAL, and CRISPR can be applied to knock out the TRAC, the T cell receptor alpha constant, and TRBC, T cell receptor beta constant plus psi, to prevent what? Why those gene are knocked out? So the ability of CAR T cells to kill tumor, no. Depletion of the programmed cell death, no. Tumor filtration, no. So they are knocked out to prevent the alloreactivity due to endogenous TCRs. Okay. Hello. Yeah, so these particular cells, yeah. So these particular technologies are being used, or these particular proteins, T cell receptor alpha and beta, has been uh, knocked out to prevent the alloreactivity, because the universal CAR T cell therapy is being used nowadays, and to avoid the alloreactivity, like if from one patient to other patient, to avoid the alloreactivity, these particular endogenous receptors are being deleted from the T cells. So what is the role of TCR in the co-stimulatory signaling domains and the CAR? They enhance the binding specificity of antibodies. They enable TCR and the co-stimulatory signaling domains. So they enhance the binding specificity of antibody. They enable the direct recognition of MHC molecule. They activate T cells. Yeah, so these co-stimulatory signal, they activate T cell. Okay. They activate the T cells. Yeah, done. So these were all of the particular questions for this particular CAR T cells therapy. Okay, these were all the things. I hope it was clear. It was a lot to take in one, but you can just go through the slides of the sir and you can read it there. If you have any confusion from this particular portion that we have talked today, you can ask me now. Is there any question? Okay, if not, then I'll conclude today's session. See you in the next class then.